Hey team, so we just wrapped up talking about osmosis. So that's the passive transport of water into or out of a cell, okay? Let's talk about now ways that cells can actively control their solute concentration, and that's through the process of active transport, hence the name. Active transport uses energy to move solutes against their concentration gradient, right? So concentration gradient, the idea that molecules will diffuse from high to low concentration. Sometimes living cells, they don't want those molecules to move in that direction. So they can use active transport to move molecules or particles in the other direction. But why is this important, right? So cells can't move water directly. They can move solutes, they can move ions, they can move other particles, other molecules, but there is no transporter for water. But because of osmosis, water will go wherever the solute concentration is, right? Water will move to the higher solute area, right? So active transport is a huge part of osmoregulation, maintaining a constant internal environment of solute concentration in the interstitial fluid, the fluid that surrounds all of our cells, right? So we can maintain a constant solute concentration in our blood through the process of osmoregulation, which requires active transport, which requires energy to move solutes against the concentration gradient, right? So we want to keep our interstitial fluid, that fluid that's between all of our cells, that's basically our plasma in our blood, we want to keep it in an isotonic environment, right? But things are changing all the time. We're eating stuff, we're dehydrated, we're drinking too much water, right? And so active transport can help control any sort of outside influences that might move the solute concentration of our interstitial fluid or our plasma into a hypo or hypertonic environment. Okay, so again, the key here is active transport is moving molecules against their concentration gradient, but that process requires energy. Okay, in order to do that, that requires energy. So we're going to use an example that you will 100% see this again once you get to an anatomy physiology series, and that is the example of the Na plus K plus ATPase transporter. Okay, this transporter is in high concentrations along the axon of neurons, and it's what allows neurons to basically function, to send electrical signals. So in order for neurons to send signals from one part of the neuron to their connection with another neuron, they need to create what we call an action potential, which is basically just an electrical signal. And they generate that electrical signal by differences in ion concentrations on either side of their membrane. Okay, so they have a resting potential, which means a difference in basically charge on their membrane that is the result of these different ion concentrations. So typically when a neuron is at rest, meaning that it's not actively firing, it's not actively sending a signal to the next neuron, we have a high concentration of sodium on one side of the membrane and a high concentration of potassium on the other side of the membrane. Okay, so high sodium concentration on one side, high potassium concentration of ions on the other side. This doesn't just naturally happen this way. These sodium potassium ions are concentrated on either side by the process of active transport. And what's going to happen is, in order to create that action potential to send that electrical signal along the neuron to activate the next neuron, we're going to swap these ion concentrations basically, and then get back to resting. So we're going to swap sodium potassium concentrations and then return them to their high sodium on the outside, high potassium on the inside. So what does that look like? So we don't need to know the minutia of this right now, but I like this example because these pumps, these active transporters are working a ton in neurons, right? So here we have sodium in blue on the outside, potassium in yellow on the inside. And as a signal is sent along the membrane of a neuron, the concentration of these ions switch. This is being shown here. right? So we see the sodium coming in, potassium going out, and then it reestablishes itself back at that resting potential where there's a large concentration of sodium on the outside and a large concentration of potassium on the inside. Okay. Again, remember, molecules want to diffuse. Even if they can't fit directly through the membrane like these ions, they still want to move down their concentration gradient. Okay, so how does the cell get back to that resting potential, right? So like after the action potential moves through and they swap, how does it get back? How does it get back to reestablishing that high concentration of sodium on the outside and that low concentration of sodium on the inside, but a high potassium concentration on the inside? Okay. That process of reestablishing that resting potential, lots of sodium on the outside, lots of potassium on the inside, is done through active transport, which requires energy. Okay, this is one of the most famous, one of the most well-known active transporters. You will definitely see this again. Okay, this is the NaK+ ATPase transporter enzyme. It's found in our neurons and it's found in lots of different cells. So when the cell is at rest, there's a large concentration of sodium on the outside, a low concentration of sodium on the inside, but a high potassium concentration on the inside. As soon as that action potential, that signal that basically activates the next neuron, is sent down, the sodium will rush in, potassium will rush out, and then the cell needs to reestablish that resting membrane potential of high sodium on the outside, low sodium on the inside, but high potassium on the inside. Okay, and so that difference in concentration is established by this enzyme, and this enzyme uses ATP to move those ions. Okay, so this is active transport using ATP to move those ions against their concentration gradient. So this enzyme, sodium is already relatively high concentration outside the cell, but it's moving more sodium outside. So we have low concentration sodium inside, high concentration sodium outside. It's moving sodium against its concentration gradient from low concentration to high concentration. Okay, so it's moving these ions in the opposite direction in which they would normally diffuse. All right, so here's kind of our overview of how this works. Okay, so active transport, moving molecules or particles or ions against their concentration gradient, requires energy. Okay, to establish that resting potential, lots of sodium on the outside, lots of potassium on the inside, that requires energy. So let's walk through how this works. All right, so we are, again, trying to move sodium out. We're trying to reestablish. Sodium had just rushed into the cell right after that action potential went through the neuron. So sodium rushed into the cell, like, yeah, move down my concentration gradient. Now, this enzyme pumps sodium back out, says, get out of here. We don't want you, okay? We need you back outside. We need you to concentrate back outside the cell. So what happens is that this enzyme kind of has two conformational changes. We call them E1 and E2, but basically what's happening in one state, that this enzyme, its active sites are facing inside the cell into the cytoplasm. And when it's facing inside the cell, when these active sites are open to the inside of the cell, right here, it is highly attracted to sodium. The chemical environment in this enzyme right now is very attractive to sodium. So these would have a negative charge to them, these active sites right here, right, in order to attract that positive sodium. All right, so sodium loads up because it's attracted to that negative active site of this enzyme. Once sodium is loaded up, that then triggers basically the phosphorylation of ATP to ADP. So we're going to break apart a molecule of ATP, adenosine triphosphate. We will break it apart into adenosine diphosphate. So we're going to remove one of those phosphate groups from ATP. And basically what that does, removing a phosphate group from ATP releases energy from that molecule, that now this enzyme can use that energy from that phosphate group that was removed from ATP to pump sodium against its concentration gradient. Okay, so We now have 3 sodium ions that are bound to this pump. We're going to move them against their concentration gradient. So outside the cell, we have a high sodium concentration. Sodium doesn't want to go that way. It wants to move with its concentration gradient, right? But nope, we want sodium high concentration. We want to increase the concentration of this ion outside the cell. So we phosphorylate an ATP into ADP, okay? Now this enzyme is charged up. It has energy that it can use. By binding this phosphate group, that actually causes a shape change to this enzyme. Anytime an enzyme's bind to something, that usually causes a shape change. And what's happening here is that this shape change is actually going to cause this enzyme to flip. So it's going to cause this enzyme to, binding that phosphate group, causes this enzyme to open up to the other side. So it causes a conformation change, which opens the enzyme to the other side. Now the enzyme is open to the outside of the cell. Okay? And that's where we want the sodium to go. We want the sodium to go on the outside of the cell. So phosphorylation leads to a shape change, which that shape change also leads to a decrease in how attractive those active sites are to sodium. Now, this shape change, sodium is not attracted to those active sites, so it's going to float off and go into the extracellular environment outside of the cell. But now when it's in this conformation with its active sites facing outside of the cell, it is highly attractive to potassium. Okay? So remember, we want to move sodium out and we want to move potassium in. So when it's in this conformation, potassium's like, hey, I like this. This is a nice comfy place for me to come in, right? It has the correct chemical environment, has the correct physical environment. The shape of it is more conducive to potassium than it is to sodium. Okay, so it has a high affinity for potassium. Once potassium binds to this active site, that binding of potassium triggers or results in the release of this phosphate group. So again, whenever something binds to an enzyme or to a protein, the shape changes usually. When the shape changes, that alters its function, right? But in this sense, we want it to function this way, right? This is how it was intended. The shape change is intended. So potassium binds to the active site. Potassium moving in from the outside fluid binds to the active site. That then triggers, once those potassiums are both bound to this active site, 2 potassiums are bound, that then results in the release of that phosphate group. Okay, so potassium binding results in a shape change, which causes this enzyme to get rid of that phosphate group, to release that phosphate group right there. And once that phosphate group is released, guess what? We get another shape change, right? Now, after that phosphate group is released, that enzyme then opens up to the inside of the cell, and that shape change also results in a change in the active site for potassium, which means that potassium now has a low affinity. It doesn't really want to be in that active site anymore. That shape change has caused a conformation change of that active site, which is no longer conducive for binding to potassium. So it releases its substrate, potassium, into the inside of the cell, into the cytoplasm, where we're trying to concentrate potassium on the inside and trying to concentrate sodium on the outside. So we're trying to pump sodium out and bring potassium in. As soon as those potassium are released to the inside of the cell, it's ready to go again. Now this active site is ready to grab onto some sodiums, right? And then we'll get a phosphate group attached to it. It'll pop back out, and then the whole thing will start over again. But the key here, the big core idea of active transport is that we are using energy to move solutes, whether it's ions or other molecules, against their concentration gradient. So against the direction that they would normally diffuse. So we typically use active transport to concentrate solutes on one side of the membrane or the other, either pumping them out or bringing them in. In this process of active transport, not only is it really important for how neurons function, it's also how our kidneys function, right? Our kidneys are moving solutes around, which also lets water move, which lets us basically remove the stuff that we want to keep from our blood, put all the waste out into the urine, basically. Okay? So active transport lets us maintain a fairly constant internal environment despite changes to the external environment. And we can use active transport for cool stuff like sending electrical signals in neurons.